All right, here we are. Thank you, everybody, for joining us for our, I think, penultimate Honors College Colloquium of the fall semester. This is our annual virus lecture by Dr. Benjamin Neuman, who's professor of biology at Texas A&M University down in College Station. Today, he will be giving us a talk entitled Learning the Unwritten Rules of Virology from Viruses. So I will be quiet. If everyone else wants to mute themselves, we will turn it over to Dr. Neuman, and then we should have some time for questions at the end. Take it away, Ben.
All right, let's look at this cute fish. Yeah, so let's talk about viruses, and we'll Christmas it up a little bit in the more depressing places because I think you've got to this time of year. So this is one of the new viruses that we published this year from that fish, which is not the puffer fish that you can eat and almost die. This puffer fish will always kill you. Just check the species before you eat your sushi, I would say. And it has a coronavirus. So what more can you want? All right, let's start our Christmas, as on every Christmas, with the Thunderdome. This is the children's play park version, which is a little more, I don't know, PG, whatever, just plain PG. We'll Christmas it up a little bit. The little tree there. There we go. All right. And so, one of the things that we've been doing this year is trying to find a way to do a universal competition for viruses because we want to figure out which one of these things is going to be the next big problem. And usually it's the one with the most, we call it fitness, but like ability to leave more of itself behind. And there we go. So the way it usually works is like this. You throw a bunch of things in. That's your P0 over here. And then with time, you'll see one of these things emerge victorious. The blue one in the top, the red one in the bottom. And then you can say, oh, okay, this was the winner. And the problem is that you've got to do the comparison for every pair of viruses or variants in the world, and there are a lot of those. So we wanted a gold standard, a way to sort of compare all these things. So we got two of these viruses. One is the SARS coronavirus you know and love, and it has a green fluorescent protein. It's called the Venus reporter in it. The other one is a SARS coronavirus, same strain actually, but with a red fluorescent protein called M. cherry. And you can put them both in there, and they mostly go to separate cells because they're duking it out in there. So these are our little competitors. And the idea is that if we have the green virus in there, and we throw in a competing virus, it may suppress some of the green virus. And so then you can just look how green it is and you can see, oh, who's winning, who's losing, and you can quantitate it. You can say, all right, my kid can beat up your kid, how much? Yeah. And so it's basically, yeah, virus Olympics. That's what we're going for here. And this is Tamina's work. So let's move myself back over. There we go. We got to see Tamina. All right. The thing we didn't expect, though, when we throw in the certain viruses that are really close to each other, yeah, they fight and one of them wins, one of them loses, and that's exactly as it should be. When we throw in two viruses that are a little bit different, they're both SARS-CoV-2, but there's about 100 mutations between these two things, then they start helping each other. And so what you're looking at here is, let's minimize me. There we go. Over on this side, this is how much of the virus green we're getting. The orange bar is how much of the virus RNA we're getting. Now we're throwing in the same amount. It's like half this virus and half the green virus. And so you get less green. That's fine. You get about half as much. But look at that. You get a little over double the amount of virus actually being produced by these two things because they don't want to fight each other. They want to help each other, which is terrible for us, but kind of interesting. Yeah. There we go. Now, this is probably related to the new understanding, and I don't know if you've seen this come up in the news or whatever, but it turns out that once you get SARS-CoV-2, you may be infected indefinitely. They can show that in various kinds of monkeys and apes, the infection will last at least a year. That's just when they stop the study. And they just did another one of these healthy human experiments where they actually gave people COVID, yeah, on purpose. And about a third of them developed one of these sustained infections where the virus hangs around essentially indefinitely. And so if you've caught it at any point, I certainly have, there may still be a little of it inside of you. The interesting or maybe bad thing over here is that if you get an infection and you knock it out right away, that's fine. And the size of the blue dots is telling you basically how much of the immune system is activated. Yeah. If you get an infection and you got to really fight it off, you'll make a pretty good response. That's right over here, the infection that they clear in a couple of days. But if you get a sustained infection, this virus is so immunosuppressive that it essentially hides. And so you make very little immune response and you just carry this thing around and it's doing minor damage, but kind of forever. Yeah. So that's not great. Yeah. And so that's one of the things we can learn. These two things are probably related, the cooperativity and the persistence, because there are all these genes in the virus, all the ones with little stars have pretty reasonable ways of shutting down your immune response, either between cells or inside of a cell. And that would help the virus that's in there. But if you think about it, that would help the virus that's trying to get into the next cell even more because a sick cell will send out messages to tell all the other cells, hey, lock the doors, there are viruses around. And so by suppressing parts of the immune system, they can cooperate without really knowing what's going on or having a brain, which is kind of neat. Yeah. But, Christmas, so put a little tree in there. Yeah. And it's all better now. All right. So this is a slime mold, and this is kind of interesting looking. Once you get a virus inside of a person or a monkey or anything else, it will start to do this. It will start to diversify into a cloud of related viruses. And yeah, that's exactly what happens. We actually use this against the virus. So one of the drugs called molnupiravir is this neat little thing. And so it'll do this. It's see the little hydrogen in a circle. It can be attached to this nitrogen or it can flip up to this nitrogen over here. But interestingly, when it does that, when it flips up to the top one, then the molnupiravir base binds really well to G. And when it flips to the bottom one, the molnupiravir base binds really well to A. So this is like a little Trojan horse. You put it into the genome, you just soak the cells in it, and your cell is smart enough to avoid it, but the virus is dumb enough and fast enough that it just puts these things right into its genome. And then once they're there, they're just like a little, blinking mutation sign. And they're coming in and they're usually replacing a C because they look most like a C to you. So yeah, that's a thing that is done. So what we tried to do is see, well, all right. And so this is Deshna from my lab. How does this affect this cooperation thing that we've seen? So we took a real cooperative virus, and so that's our blue bar over here. So we're comparing that to green all by itself. This is how much green you get out. It's almost like 3 times as much because the blue virus helps the green virus so much. Yeah. And with the red, it's about the same. It's about 3 times as much red coming out because they're helping each other. But if you take this same virus and you take the same amount of the same virus and you spread it out a little bit, so you make these random mutations in it, then what you end up with is a virus that doesn't help quite as much. Now, this is the same. It's not that we're losing viruses that are not able to infect because we're controlling for the same amount of infectious virus in there in all of these. And so this is like kind of like a narrow population, and it's super helpful. The medium broad population, a little less helpful. And the very broad population here is actually almost competing now with the original virus. So in a weird way, it's kind of saying that diversity leads to competition in these viruses. Like they'll work together if there are only a few different types. But when you start to get a swarm, then it no longer works well with some of the other strains. So like the swarm is its own thing now. And I think that's kind of interesting. And Christmas. There we go. All right. So This is neat. This is probably your favorite pastime. It's not mine, but deer in Texas are big business, and there are a lot of people that are breeding for interesting antlers, just like way too many points, like this guy and some others that I've seen. Just fantastic. So anyway, these deer are expensive. They are basically we've made the forest spirit from Princess Mononoke. We just got to work on the face a little bit, but everything else is there now. And so at one of these farms where they're growing these deer, we've got a veterinary group that goes out and they sort of swab the animals to try and check and see, all right, what's going on? Is this virus going to spread to deer and then spread back to people? Something like that. And unlike the deer in the forest, these guys are all in a nice little fence and you can get to them and swab them and they come up and get their dinner from a little tray. It's, yeah, much easier. So this is Francisco, and he's the lead on this one. And so what Francisco and his group found is that in a population of deer in Texas, oh sure, let's let that person in. There was a SARS-CoV-2, and that's neat. And there were actually a whole bunch of them in this herd of deer. So all these ODs, these are deer viruses. And so we were able to isolate a bunch of these things. We were able to sequence these things. And compared to the human virus that's circulating then, so these are from around the beginning of December. And if you look, your beginning of December viruses are right about up here. And so every step is a mutation. So they're probably catching something right about like that. And then when you get down here, you're into sort of January or February the following year. So this is the normal rate of virus mutation. It looks like these guys made a lot of changes really fast, and they did. Uh, so if you compare it to normal human SARS-CoV-2, it's a straight line. This virus has been making exactly 31 changes per year, every year, and probably will go on doing that until long after you and I have died of COVID. Yeah. But this virus made half a year's worth of changes in 1 or maybe 2 months, but probably one. And that's kind of alarming. So we're trying to look at, all right, why is this? And you can look at the mutations and they kind of tell you. So the deer viruses have a bunch of changes that are a C turning into a T, and a bunch of changes where an A turns into a G. And those are the only two changes out of all the changes that are possible that we actually have a good mechanism for. So inside your cells, one of the things that cells can do to become more virus resistant is they turn on this thing called ADAR and this thing called APOBEC. And both of those force the virus to mutate. They force little changes to happen in the virus, kind of at random, but it's always C's to U's, just like randomly, or A's to G's, essentially, randomly. And so about three-fourths of the mutations that we see in the white-tailed deer look like they're coming from these two pretty normal mechanisms. But these are happening in the deer and they're not happening in the human. We think that's because these viruses are not as immunosuppressive in the deer. So the deer do a better job at clearing them out, and this is probably the signature of it. They're mutating the heck out of these viruses while they're there, really beating up on them. And so if you're asking, well, all right, normally mutations are bad. You probably remember that from other lectures. And so what does this do to the virus? And so the colorful ones are the deer viruses. The gray ones are most of the human strains that were around at the same time, like December 2023. And you can see if we're looking at 24 hours, yeah, the human origin strains tend to grow a little better than the deer origin strains, but by 48 hours, they have completely caught up. So they're just slightly slow in the early stages, and then they do fine. And kind of the problem there is that if you get these things going out to animals where they are hyper mutated and then they come back to humans, it's not that the virus is weakened or unable to grow or anything like that. It's just that it's now got so many changes that your body may not recognize it anymore and it may be able to infect where it couldn't have otherwise. So, yeah, I don't know. Here's a big Christmas tree. Yeah. All right. Shi Ching, yes, is a good chemist here at Temu, and he's been trying to come up with a different way to attack these guys. So we're going to shift over to the attack. Yeah, the viruses have been beating us up so far. And it's kind of a neat little mechanism. So he takes a inhibitor, which is the red part over here, keeping the Christmas colors basically. And so that thing is going to bind to part of the virus. And then he takes it's called a pomalidomide molecule. That's our little green thing hanging off over here. And it's going to be a thing that'll bind to cereblon, which is the CRBN. And that is a protein that's involved in this system. So, ubiquitin system is basically the cell's way of recycling proteins it doesn't want anymore. And so the little ubiquitin's the little yellow dot, and it gets passed to one protein, then to another protein, just like a good basketball team. And then they interact with this other one, and then together they're going to stick it onto the protein, and then the gray protein is going to get destroyed. Yeah. So that is the idea. You put something in that'll stick to your virus. You put something in that'll stick to the cell's basically garbage disposal. That's the proteasome. And the idea is that, yeah, you take care of two problems at the same time. The virus can't grow, and the pieces of the virus get destroyed. And overall, they're kind of mid. Yeah, you can put in very large concentrations and you can actually suppress the virus pretty well. But they have to be right about at the level where they would start to make the cells sick. So it may not be something that has a lot of use in the clinic by itself. And it seems to stack well with some of the other drugs. These are some of the other things that are approved treatments. But, and this was unexpected, if we take two of these that hit different parts of the virus with their little virus destroying powers individually, we're teaming them up with nirmatrelvir. This is the active ingredient in Paxlovid. Yeah, brought to you by Pfizer. And so the G is one of these and the X is one of these. So N is nirmatrelvir. G is one of our little, they call them Protex. And so you can see on their own, they don't do much. Like if this is how much you have to put in, and this axis is how much virus is growing. And you see we're not really making much of a dent. But put in the G and the R and the X all together with your nirmatrelvir, and then all of a sudden you can completely flatline this virus. And it looks like you may actually be able to cure an infected cell with this by physically destroying all of the virus that's in there. And that would potentially be a thing that may be able to combat this sort of long-term SARS-CoV-2 infection that it seems like probably a lot of us have right now. Yeah. All right. So on to lasers, because lasers are fun. And this is Nara, and Nara's very smart. I don't know, I didn't know this at the time, but Narangarel Altangarel is one heck of a Mongolian name. About as Mongolian as you get. And she's fantastic. She's the one that's leading the laser aim of what we do. And what you see at the top is what everybody else is seeing. So you shoot a laser at a protein, and you can get a little bit of structure information. You get little bumps, you get little dips. Very nice. Then, if you don't do anything and you just shoot the laser at the protein again or shoot it for longer to get a little more signal, what you'll see is that it starts to change. Like, look at this peak, it's sort of flattened out. And this peak wasn't even there, and it starts to appear. Same thing for this one. This peak was really tall and nice, and it just gets worn down to a nub, which means that basically your laser is destroying whatever it is you're looking at. And so her tool, the goal of what we're doing is to try and figure out when things are binding to the virus to try and make better antiviral drugs. Our giant invention on this, and we've actually patented this, it's so dumb. Yeah. Is that we refrigerate the sample. Yeah, that's it. That's the patentable thing that for some reason nobody else has thought of. I don't know. Anyway, with cooling, you can shoot the same sample 4 times. You get essentially the same spectrum back out. None of this stuff. Even though the same amount of laser energy is going into the sample. Kind of cool. So we're trying to use this for something new. So we've got, so every protein's made of these little things called amino acids. That's all the little colored dots over here. And there's 20 different ones that you get in proteins, and that's fine. Each one of them has its own little spectrum when you shoot the laser at it, Raman spectrum. And so the question was, if we know the spectrum of all the little parts that make up a protein, and this, and we're cheating and we know which protein we're looking at, can we predict what the spectrum would be for the whole protein? And then, if we can do that, if we take an unknown protein, can we just look at it and say, oh, we can tell, it's got 5 prolines, that's the pro thing here, and 3 cysteines. Can we actually estimate the amino acid composition of one of these things from the way it looks, essentially, to the laser? Now, the one other missing piece here was that we're looking at loose amino acids, and loose amino acids have 2 free ends that aren't doing anything. Whereas in a protein, the amino acids are all linked together in a big long chain like this. And so only there's one little free end over here and one little free end over here, and all the rest of those ends are linked up. And so we had to look at, okay, let's just take one amino acid. This is alanine right here. And we'll look at one copy. And so one copy gives you all of these peaks. And then what if we get 5 copies of the same thing? Let's look at that. And so it has a lot of the same peaks, but there are some extra peaks that go away or appear. And so you get the appearance of the peaks that are actually the joins between these things, and you get disappearing ends, essentially. And so you can map that out. And so with that information, you can now go in and actually do this. So here's insulin, and insulin's got 1 alanine and 1 arginine and 3 asparagines. And so you just multiply the spectrum for asparagine times 3, and you add these things all together, and you'll end up with these little peaks at the end. And that turns out to be a real good match for the actual spectrum of insulin. There is another component of this, which is that there's this structure. So these little coily bits are called alpha helices, and the little sort of flat ribbons are beta strands in a beta sheet. And you can actually either observe these or you can infer these from the Raman signal. And the difference between the sort of calculated and real is something like 2% for most proteins. And we can actually get a good structure estimation to about 4%. Yeah, variation. And that's something else. That's a thing we couldn't do before. This, we think, may have some use in therapeutics, like when somebody makes a jar of RNA vaccine, for example, and they want to send it through the mail, and they want to check that it didn't get, left on the at the airport on the tarmac in Houston or something like that. You can check and see if it's still intact. And you can see if there have been any changes to the structure of these things or any changes to the signal from the amino acids from these things. It ought to be a real useful tool for that because it's a pretty hard thing to do at the moment. And it takes about a minute with this technique. So it's kind of cool. All right, last bit. And you'll like the next thing that comes up. Not this one, but the one after. You come here to hear about new viruses, I would assume. Otherwise, why are you here? And so new viruses are going to come from what's called next generation sequencing, which is where you take all the RNA from inside an entire organism and you shred it up into random sized pieces, just like breaking up puzzles, and you mix them all together, just like you would never want to do with a puzzle. And then you can read them all out, and then you can use a computer to reassemble all those bits into what they may have started out as. And that's essentially the bioinformatics thing. So we're working with Chris Lauber over in Germany, and he has invented these two, they're called pipelines, but bioinformatics pipelines, Virus Hunter and Virus Gatherer, that use two pretty clever ways to find viruses from among these little scattered tiny bits of things that are inside of cells. And he's really good at it. And we've been helping out with this. And kind of the secret sauce that makes it work is that he's not just using like trying to match, like taking a virus and trying to match it to another virus, because that's okay when your viruses are close. And when your viruses are not so close, it's not as good. He's using a thing called a hidden Markov model, which is more sophisticated. It's almost like the way an AI would work. Some similar stuff. But anyway, he gets a lot more good hits from this than the other competing method. And hey, if you want to try it yourself, it's available on GitHub. There's the link. Go out and get it. Why not? And so the summary, I was like counting these with my finger on the screen about an hour ago, so I may be off by one or two, but all of these sequences, we have looked at the same data. And so seven have come in from, other people just sending you their cool virus and saying, hey, can we put this on a tree, build it into something? And we say, sure. 38 are from the oldest method, and a lot of those are ones that I've pulled out. 16 are from the latest, greatest AI method, and that's by Seratus, is the name of it. But surprisingly, it doesn't hit as many things as you think it might. And Virus Hunter and Virus Gatherer are just absolutely cleaning up here. These are only viruses related to coronaviruses. It works just as well. Actually, all these things work proportionately on other viruses, but this is an absolute game changer. The only problem is that you get out partial viruses from this, and that's where this guy comes in. Just you figure out what kind of errors you have in these viruses, and you go back in and you tinker around with the computer. So I argue with computers. That's essentially my role in bioinformatics. But yeah, then you can build out the rest of the viruses, and you get nice complete things. This is from our new paper. Hopefully, it's going to be accepted this week. And what we found is that viruses like coronaviruses are unusually diverse. They're just absolute Wild West. So, to compare this, if you take the largest group of viruses, they're things related to polio, largest group of RNA viruses. Every single one of them, they do a lot of stuff that's different, but every single one of them actually has its RNA copying unit set up in exactly the same way. It's always one protein with three things in a certain order and no variation tolerated whatsoever. Coronaviruses do all these crazy things. So this is seven modes, but if you count the A's and B's, it's nine submodes, nine ways of making a genome that basically you don't get in any other virus family. Coronaviruses and their relatives are experimenters, and that's probably a lot of what makes them concerning, I would say. All right, so this isn't reading class. You read it, yeah. Tell me when you're done, and then we can talk about it. All right, I'm going to assume you read fast. Yeah. Okay. So, also with a couple other people at the university, we tried to expand this. So if you're playing around these little tiny sequence bits, there's other stuff you can do. And so one of these unanswered questions was, well, people have been reading out lots of bits of DNA and RNA for, I don't know, 30, 40 years, and they've been building up in these databases. Nobody has any idea what's in there. And so the task was, can you catalog all of the sequence information that humanity has collected? And specifically, can you do that and figure out where are the gaps? Like, where is there a group of animals, particularly a group of animals that is important to us for some reason, where we don't have a lot of information? Because, those would be good things to fill in those particular gaps. And so here we go. So what we did was to download first all of taxonomy. Taxonomy is like kingdom, family, phylum, whatever, genus, species, all that stuff for everything that's known. So all the bugs, all the insects, all the eukaryotes, all the things with a nucleus ended up in here. And we had to actually modify this because plant taxonomy is done a little differently and it's not stored in the same place as animal taxonomy for weird old reasons. But yeah, we built a kind of a unified taxonomy downloaded not all the sequences, but all of the sequence information, all the metadata, they call it, like the little words at the top of the file, to figure out what these files are and how many of them there are, and ran them through counters and stuff like that. And yeah, we can then build this little tool which works online. It can access this great big spreadsheet and it can tell you how we're doing at certain groups. So everything that's on the screen is an arachnid, and the lighter the color, the more sequence we have compared to the number of organisms that are in there. So, like looking down here, this bright yellow one, ticks, we're doing great on ticks. We got a lot of sequence coverage on ticks. Could be better, but not as much of a problem. Spiders were really bad until 2 years ago when this giant study came out and got 1,000 spiders, got a whole bunch of genes from each one, really helped with that. So good job, spider paper people. I think it was out of Japan. Yeah. But then look, whip scorpions get no love whatsoever. Scorpiones, yeah, you can figure out who those are. The regular scorpions. And yeah, basically all these other arachnid groups, including the harvestmen. That's the, I don't know, daddy long legs, the one with real long legs and a little round body like a bread roll. They're just adorable. You'd want to keep them as pets, I think. But they're very poorly sequenced at the moment. And so then to figure out whether or not these things are important and attach like an importance value into this same program, we downloaded all of the scientific literature that's existed, like papers going back to the 1700s right through today. And this is actually a good use of these large language models, like the thing behind ChatGPT. And so if you're doing bioinformatics and you can figure out how to train these things, that is a very useful skill right now. Because, yeah, in the matter of about 6 or 8 months, downloaded, digested everything, pulled out all the names of different kinds of animals and plants and bugs and whatnot, and then matched those to some keywords. And keywords like, crops or pest or medicine or poison, stuff like that. And so, for example, like if you look at the word agriculture, all the little hits that come up, you got about this much, the big brown wedge, which are things, plants that we grow for food. And the pink wedge is mostly animals that are pests of that food. And so these are the things we write about. But this is like a way to figure out the sum of human knowledge about animals and plants, essentially. It's kind of neat. And so this is where we'll leave it, kind of the last thing. These are just different kinds of things that people sequence. Like if you're only going to sequence one thing from an animal, a lot of times it's going to be either a 16S or a 12S or a COI. Those are pretty common. But it's different groups, like ITSs, I think they tend to go for in fungi, or maybe that's plants. I can't remember. But most important thing, GenBank is American. ENA is the European Nucleotide Archive. And if you look who's ahead on all these categories right now, we went through both of these. Where we are. Yeah, that's right. That's the message of today, that the US, yeah, number one in storing and acquiring genetic data. At least for now, Europe's really close, but whatever. And Christmas as well. Yeah. All right. Thanks very much. If there's anything I can help with, I am happy to help. Yeah, that's it.
Thank you, obviously, Dr. Neuman. We also appreciate the shout out to the grand old US of A. Well done. State of Texas approves. And with that, we will open it up to questions. I will. I mean, we're a cozy enough group that you are welcome to unmute and ask a question, or you can put it in the chat, and I will dramatically read it for you. However you want to do this. But I'll start with just one that from my deep ignorance of virtually everything you talked about aside from the American flag, I recognized that, and the tree. I got that too. So what do you think accounts? I mean, or I can just ask what accounts for the variability or the ability to kind of change so frequently, so often in coronaviruses specifically? Like what? Yeah, to the layman among Among them, I, of course, know.
That's fine. We'll dumb it down for the rest of them, shall we?
Yeah, for those people, those other people who, yeah, aren't medieval historians and thus know.
Yeah, obviously, obviously. Yeah, I would say we're more complex in the sense that we've got about 20,000 genes and maybe 50,000 forms of those genes when you take them all into account. And there's probably half of those that if you knock them out, you either get real sick or you're dead. And a virus will often have like 10 genes. And yeah, so there are fewer moving parts, and the moving parts have been evolving for all of time in a way that's just really fast. And so they make 1,000 children. I don't know. Actually, I do know exactly how many kids you have. Yeah. And you got a ways to go. Yeah. But they'll do that every couple of hours, 1,000 new little offspring. And 99.99% of those things never make it in the world. So have more kids, pick up more mutations, throw them out. One or two of them will make it, and you can call yourself a success. That's the virus way. Yeah.
Here we go. Win with numbers.
Mhm.
Okay, other questions? Who else wants to jump in?
I was kind of interested in your technology where you're looking at using a laser to read proteins and the components of proteins. I think at one point you started talking about how when there are slightly different shapes, it gives you different readings. So would that work for things like prions and stuff like that, where it's still the same components, but it's folded differently?
Yeah. And the one fold kills you and the other one doesn't. Yeah.
Yeah.
Yeah. And right now, the biggest problem that the deer breeders are facing, at least based on my intimate firsthand knowledge, is that if you want to sell or move a deer, you have to go through a test for a prion disease, this chronic wasting disease. And I think the test is somewhere between 100 and 200 bucks, and it takes a while. And so you lose all the profit that you were going to make off of that. And a lot of the deer breeders have shut down because of that. Yeah,
Hmm.
this is a minute and should just work. We were trying to get a hold of some of that protein because that would have been a really good one. One of the others that we looked at, though, is one of these ones that forms like little amyloid plaques. And you could actually watch it over time accumulating, like changing to the bad structure that kills you. It was kind of neat. Yeah.
Because
Yeah,
that has a lot of medical applications because the amyloid plaques, I think, are what builds up when you have dementia,
for
right?
sure.
Yeah. And
Yeah.
then, are you familiar with IgA nephropathy?
Mm-mm.
Okay, so I could be incorrect about this because it's been a minute since I've read about it,
I
but
won't know. Yeah.
I know it's a buildup of your IgA protein in the kidneys, but I can't remember if it's because of a misfolding where it starts to actually build up.
Probably. Yeah.
But if you mostly it gets so bad with this disease because there's no good way to detect it before you're like peeing blood all the time.
Ugh.
Yeah. But if you could find a way to differentiate proteins in tests, especially if you just have extra protein in your urine before you start seeing more severe signs, that would be a great tool for medical providers.
It's potentially on the table, but I'm not ready to shoot myself with this laser. It is an infrared laser, and they're the same kind they use at the dentist to sort of clean under your gums and just murder everything in there except your cells. Yeah.
Yeah. I
So
was thinking
theoretically, yeah.
if you got a urine sample, I think you could test for it that way because it builds
Oh,
up
yeah.
in your kidneys.
Then that would be easy. Yeah. Urine is either like urea or urea plus something, and you would be able to tell. Yeah, for
Yeah.
sure. Yeah.
Oh, okay.
Okay, thank you, Caitlin. Um, any other questions from anybody? Anyone else want to jump in on this? Not all at once. Kathy?
Oh, I too will lay my ignorance right out here on the table. Um, you mentioned two things I understand: deer, Christmas. I got that. Um, one of my doctor cousins, of course, this is a big deer hunting, world we're in. one of my doctor cousins says, don't eat the deer meat because of the wasting disease, the ticks, the yada yada. So from a person who knows, what would you say?
There's a couple studies that came out, I think, this year, and they tried to transmit that chronic wasting disease, which is a protein misfolding thing, like we were talking about. Um, and they could transmit it from deer to other deer. They could barely transmit it from deer to elk, and they couldn't really transmit it any further than that. It looks like it may be different enough that it's probably okay, but I don't know. I would burn it to a cinder anyway. If you cook it really like too much, yeah, like they tell you not to cook it, that does appear to misfold the protein enough that you're okay.
Jerky. Make yourself some jerky.
Sure, go for it. I
I,
think.
Jerky's a great idea.
Yeah, yeah,
That's a good takeaway from this talk. Make deer.
yeah, yeah. I'm happy with that. Yeah.
Yeah. Any other questions for Dr. Neuman before we?
Here's my other
Oh,
ignorant.
sorry, Kathy. Go ahead.
I'm just full of them today.
Love it.
I have quarterly labs. I'm a multiple myeloma survivor. I have quarterly labs, and they test for IgG, IgA, and IgM. Now, is that
Yeah, those are all antibodies.
okay?
IgM is like the first antibody that you make. So that's usually like you have something going on right now. And then IgG is like you've been seeing something for a while, and this is your, the one you're going to stick with version. I'm not sure what they're looking for antibodies against. If it's antibodies against a certain protein that's only made in the cancer, that would make sense. Because like you could always find the cancer if you can like dig it out of the human body or like dissolve the human body and just, leave that part. But the trick is to do that while the human is still, alive and not as inconvenienced. And
And
so the
this
antibodies, I think you'd be able to pick that up in the blood. I would guess that's what they're doing.
yeah,
But
they just take labs, and because that's how they find the myeloma. They look at these indicators, and but I did see IgA on your chart, and I got so
yeah,
excited. That
yeah.
and the Christmas tree and the deer. I'm sorry, I had three things. Okay, I knew
That's
three.
the antibody in snot. That's why we had it on there because yeah, snot's good stuff. Yeah.
I feel so well. Thank you. My
You're
labs are
very
all
welcome.
normal.
Awesome. Well, that is good to hear. And an important safety tip from Dr. Neuman: do not dissolve the human body to eliminate cancer unless you can guarantee the human body survives.
Well, yeah.
Any last questions? So I guess why biologists are not quite the same as doctors. Yeah, different interests.
At least
Kate.
10% the same. Yeah.
Question. So if it turns out, which this may be just understandable, but or makes sense, but if someone's going to get sick with another virus, like say flu or viral pneumonia or something like that, and they do have the COVID virus that is sustained for a while, or forever in their body, are they going to get way worse, or their sickness is going to last for much longer? That's been debated kind of medically with some people, but
The papers showing it hangs around for a year are just coming out now because they were baking for a while. We don't have an answer on that, but if the virus is constantly there and the virus is constantly immunosuppressive, I don't see how it couldn't make other things
right,
worse. Yeah.
right.
Yeah, it just seems like that has to happen. Yeah.
Well, that's like my uncle, for example, he had pneumonia and he was in the hospital forever. I mean, they were just loading with antibiotics and all this, and they asked if he had had COVID before. And anyway, another question I had, so when you're thinking about treating this, again, this may be another silly question, but when you showed the chart about like how they all start to work together and this is the
Yeah.
one. So
Yeah.
when
A
target,
beautiful Christmas story. Yeah.
yeah, thinking about like mass vaccines and that kind of stuff, would you target the one that's like the culprit or the one that's already developed from all their friends gathering around?
Yeah, we're just figuring out which ones help each other, and we don't have good like boundaries.
Okay.
And you don't ever get a choice in which virus you're exposed to because you're either,
Right.
wearing a mask or not.
Yeah, good point. Yeah.
Yeah, yeah. But yeah, I think that could influence vaccine policy if we continue to have a vaccine policy in the next 4 years. Yeah. Just because, yeah, if there is one that's making everything worse, yeah. Yeah. It would only apply when a person has both of them in their body at the same time, but you would want to limit that as far as possible because then you get recombinants and all sorts of bad
Sure.
stuff.
Right. Right. Okay, one last question. So the one with the ticks and the spiders, are we ever concerned about mosquitoes? Thinking about other kind of blood.
We're not doing bad on mosquitoes. Yeah, in terms of genetic sequences, yeah, they're a big enough, annoying enough problem that yeah, they're pretty well covered. But there's loads of other things that aren't, and like viruses bounce between all these things, and most of these things we don't really understand what it is they're, doing and how they fit into everything. So
Right.
no, there's a ton of stuff that's not known. Yeah,
Those are they're so bad in East Texas, the mosquitoes. So
I remember. Yeah, they're not doing anything good. Yeah. Hmm.
thank you. That was all I had.
All right, any last questions for Dr. Newman? All right, seeing none, I think you're good for another year, Ben. We will be in touch, obviously, again next year, for your eternal lifetime commitment to offer us a December honors colloquium on. Doesn't have to be on viruses, though. I guess it is in the title. So, but no, thank you once again. It's always good to hear from you and very interesting stuff. I will go ahead and end the recording and I'll get it posted to YouTube for the folks who want to watch it later. And yeah,
Over and over.
watch it again. Someone might, or for people who missed it, which is probably the more likely audience, I guess. But thank you, folks.